peripheral proteins, and we've got the hydrophobically buried integral membrane proteins. There's a lot of variety in these. If we look at the integral membrane proteins, we've got some like this one that just have a single pass going through the membrane. So remember, proteins are polymers of amino acids, so they're long. They're long strands of polypeptide chain that wrap up into various shapes. A lot of these membrane proteins will start with one end on the inside, the other end on the outside. And in the middle, they're anchored because they've got a long string in this segment that's where the amino acids have hydrophobic side chains. So these guys don't want to interact with water. In a normal protein inside the cell, they would cluster so that they're buried away from the water. But here they can live happily inside the hydrophobic core of the membrane. So this is like an anchor, and they'll be part on the outside, part on the inside.
So they're the same amino acids, but they just have different side chains?
Yes. The 20 different amino acids include quite a few that have hydrocarbon side chains. And these are hydrophobic. So they don't want to interact with water, things like isoleucine and leucine.
I guess I'm seeing valine.
Valine is a branched hydrocarbon, nothing but CH2.
Because I'm seeing them on both sides, I guess that's what I was confused about.
Out here, you mean?
Yeah.
Oh, don't worry so much about that. We'll talk about that. That's a good observational. When we talk about proteins, we'll talk about that.
Oh.
What that probably means is some of this is coiled up to bury those. So it may be more globular here, more globular there.
Okay.
But the ones in the center here, you don't see polar stuff here.
Yeah,
for
okay.
that. And outside you see ones with carbohydrates. So this is a way to hang stuff off the outside of the cell and anchor it. Some of these systems are more complicated. It will provide a way for communicating stuff from the outside of the cell to the inside of the cell. This particular simple example with a single transmembrane segment, just one pass through the membrane, this example happens to be glycophorin in the red blood cell membrane. It's the one that carries the blood type carbohydrates on the outside. But there are more complicated ones where one protein will take multiple passes through the membrane. There's a whole set of them that have 7 transmembrane segments that are often involved in hormone systems. This is a multiple pass one. Same kind of deal as the last example, except it goes back and forth, back and forth. They'll come up on one side and then dive back down and then cut through again, really anchoring it. Why would you have stuff like this? Just in a general sense, if you're going to build some kind of gate mechanism that's going to let something from the outside come into the inside, you need to provide something like a pipe structure. And to build that, you need lots of protein segments to kind of make the wall of this pipe or gate structure. This one is bacterial rhodopsin. There's a good crystal structure on that. That's mainly why I picked it. It's bacterial membranes. It has a rhodopsin molecule in here. Captures light like the molecule in our eyes. It's photosensitive. It captures the light, uses the energy to pump protons out. It's part of its way of making ATP. But it'll pump protons across the membrane in that little channel.